Okay. Okay, thank you. Welcome to this presentation of LIPUM. And before I start, I think I should mention to you that we are not listed on any stock market. So if you're looking for the buy button, you won't find us there. And as mentioned here in the presentation, we have our roots in the city of Umeå and Umeå University and research. And the story about LIPUM goes around about 25 years back. Professor Olle Hernell, professor in pediatrics, he was studying nutrition in breast milk for newborn children. And then they discovered a protein called bile salt stimulated lipase, BSSL. And it was clear that this protein is very important for fat digestion in early infancy state. But while they were doing these studies, they also noticed that the protein was present in the bloodstream. And it shouldn't be in the bloodstream, so they couldn't really explain why is it in the blood. And maybe it was secreted from the liver, they thought. So they started to study patients and looking into the liver. Do they find BSSL protein? In fact, they did. And in particular, they found it in one patient that had the disease called fatty liver. And fatty liver is a sort of inflammation. So suddenly they realized and they discovered that maybe this protein has importance for the outcome and promotion of inflammatory conditions. So one could say that they were actually in pediatrics field interested in the nutrition of newborn children, but they had an orthogonal discovery that actually this protein has importance for inflammatory conditions. So they embarked in to study this more in detail, and because of the research, they had access to so-called knockout mice, which means that are mice that are deficient from the BSSL protein. They are genetically modified, so they cannot form this protein in the body. And they ran a trial where these mice, the knockouts, were induced by arthritis. And they used a very well-established arthritis model called collagen-induced arthritis. And parallel, as reference, they did the same experiment on mice that are wild type, that have BSSL in the body. And as you can see on this slide, there was a significant difference between the effect. The wild-type mice were seriously ill and got the arthritis, while the BSSL-deficient mice were unaffected. And arthritis and many inflammatory diseases, they are ambiguous in the presentation. The diagnosis is made by specialists, rheumatologists, that they measure how affected a joint is, for instance, and this is typically measured in a score. So what you will see as response here is the score in terms of arthritis. This model is representative for a disease called rheumatoid arthritis, or in Swedish, ledgångsreumatism, and that affects around about 1% of the global population. Later on, a similar study was done on the knockout mice and the wild-type mice in a test that is used for inflammatory bowel disease. And as you can see, the wild-type was significantly more affected than the knockouts. So the question was, can we stop inflammation by inhibiting the effect of this protein BSSL? And a lot of experiments followed. First of all, it was tested administration of an antibody that could block the BSSL protein. And regardless of the way the antibody was administered, the mice that got the antibody was significantly less affected than the control group. Also, the effect of different dosing was studied. So when the dose of the experimental antibody was increased, more BSSL protein was bound, and less affected was the test subject. And as you can see in this image here, those that get the highest dose of the antibody are more or less unaffected compared to the control group. Later on, samples were studied from real patients, and that's very much thanks to the university and the advisory. We have Professor Solbritt Rantaper Dahlqvist have samples from real life patients. And what these graphs shows is that depending on the score of the disease, the concentration of the BSSL protein varies in the bloodstream or measured in the serum. It could also be seen that when these patients got a medication, a Remicade, that's a TNF-alpha inhibitor, in the mid graph, when they got the medication, also the concentration of the biomarker BSSL decreased. And to the right-hand side, even more interesting, that during relapse, when the disease is active and the intensity is high, then the concentration of the BSSL protein is increased. And in terms of mechanisms then, the scientists and the founders of LIPUM, they found that the BSSL protein is in the neutrophils. And when it's released, it binds to monocytes. We're talking about white blood cells here. So it binds to the monocytes and stimulates the monocytes to migrate to the place of the inflammation, like in a joint. And then it sustains and maintains the inflammation in the joint. Okay, so then obviously we aim for a product that will bind the BSSL protein, and that is a therapeutic antibody that will bind the protein and inhibit this mechanism that promotes the inflammation. And this constitutes a completely new mode of action for treatment of inflammatory diseases. And being different, it also likely that this mechanism will not be an immunosuppressant mechanism. Most of the other drugs used, the most popular drugs used for treatment of inflammatory diseases also suppress the immune system. But as far as we have seen, this doesn't. And one evidence is that we have knockout mice, which means we have mice that don't have the protein, and they are just as healthy as any wild-type mice. So we foresee no relatively significant negative effect of blocking the protein by an antibody. Yeah, as mentioned, rheumatoid arthritis affects around about 1% of the global population. That means that in this market, there are a number of blockbuster drugs. And in particular then, the biologicals. The biologicals that are targeting the TNF-alpha. And also the prescription trend is to use these more sophisticated biologics, even though they're expensive, but they are used more early in the treatment cycle. Okay, isn't that good enough then? Do we need anything else? Yeah, there is one major problem with these. These are suppressing the immune system. That means that the patient becomes more sensible for secondary infections. That means also that you cannot combine two immunosuppressant drugs without increasing this risk for secondary infections by the patients. Other problems is that round about 1/3 of the patients are non-responders, so they have no effect. Another third actually loses its effect in a time of 3 to 5 years. The prices for these blockbusters are very high, still very high. And this, of course, also attracts the interest for biosimilars. But despite the fact that biosimilars are on the market, the prices haven't dropped that much anyway, because there are very strict registration conditions on biosimilars. And according to statistics from US health insurance reports, the prices for these TNF-alpha inhibitors are around about $30,000 to $50,000 US per year treated. But there are exceptions also, and one exception is for systemic juvenile idiopathic arthritis, where you can see that we have almost $400,000 in treatment cost for 1 year. And then talking about this then, juvenile idiopathic arthritis that we focus on in the first instance is a rare disease. It's a rare disease that affects children below 16 years of age. And there are 337,000 patients having the disease in the US, Europe, and Japan. And if we look into the treatment tree, you know, when you go to a doctor, the first thing you get a pill, and that doesn't work, you get a stronger pill. And then you enter in the treatment tree to a level where you actually get something that helps you. And it's exactly the same for the patients having inflammation. But in the field of juvenile arthritis, that is a rare disease, it has been very much up to the rheumatologists to do off-label prescriptions to challenge the disease in children. And that is important because the children are affected by a way that they get stiff joints, they get rash, they try to hide that they actually have a disease, and they also sometimes have to stay at home from school, and that affects, of course, the children, and the society cost becomes high. But also in some cases it becomes a lifelong handicap where they need repeated surgery to correct joints and correct bones that were affected by the relapse periods. So what we do, LIPUM, in the first instance, we focus on the fourth line treatment where we have around about 20,000 patients. Because on this level, the children, there are no available medication for them recommended. One could say that there were in 2011 and 2013 were new guidelines. There is a new guideline coming now, but most of these guidelines will actually recommend RA drugs that were developed for grown-up people, adult people. They will actually be recommended also for the children. And with the effect that they also get immunosuppressant drugs that can actually affect their childhood. So we have come up with a strategy for LIPUM that we will have focus on an in-house development on the indication juvenile arthritis, where we expect to get an orphan drug designation, other benefits from the society that will help us to get faster into clinical trials. The demands on the clinical trials will be less, and we have in-house, around the founders in the company, we have the competence in the field of pediatrics. In parallel, we see a huge opportunity in the field of rheumatoid arthritis, but there the requirements on the number of patients in clinical trials and so are so large that we will absolutely need a partner and do an licensing deal to get into the market. While talking about these two indications, it's also clear because of the very different mechanism of action, and also based on results we've seen from real patient samples, patients having psoriasis arthritis or inflammatory bowel disease, we see that our mechanism probably have impact on a wide range of diseases, going from the autoimmune type of disease to autoinflammatory disease. And this, of course, need to be explored more in detail in the next years. Yeah, well, we are not listed. That means that there are 9 shareholders in the company. The founders still have the majority, and Almi Invest, and then there are 5 other private individuals that are shareholders. We'll look a little bit more on some of them here. So this is the management and the board of LIPUM. Actually, we have our chairman, Ulf Björklund, here today, and Ulf has a very long experience in management of pharmaceutical companies. Still, 2 of the founders are on the board, Professor Olle Hernell and Professor Lennart Lundberg, and me and Dr. Susanne Lindqvist are doing the management. In addition, we have a very advanced scientific advisory board, and of course we have also established a number of well-reputed and competent counselors. And it's very much worth to mention here that already in 2015, the academic people here at the Umeå University, they got a collaboration with the Science for Life Laboratory, SciLife Lab, and that has been, of course, invaluable for us in our development of the candidate drug and the understanding of the mechanism. Our timetable now is that we are just in the process to select the candidate drug. We have actually selected it. One turned out to be a couple of candidates, but we will very soon validate these, and it's also a little bit depending on the IPR situation. We want to test a number, but we will have it in very soon. We will decide it for one that we will start production, the CMC work, and then we aim to have clinical trials to start in 2021. Yeah, well, we have very good news today. We received support from the European Union this summer. That's SME instrument and a grant of 2.2 million euro. We announced that today. And just a couple of months ago, we had a grant from SweLife for Better Health project, 1 million Swedish krona. And before that, we had support from the European Union Step 1. And even before that, NovoSeed, that Novo Nordisk pre-seed grant, was given to the researcher of LIPUM to get it moving. And all the way through, we've been supported by the Umeå Biotech Incubator that actually, beside mainly not financial support, but management support, and that is also very valuable for the company. Private investors or money invested in 2016, Almi Invest invested 2.5 million, and in the late last year, the management, the founders, and some business angels invested another 3 million. But as you know, we need more money to speed up, do the job faster, and to explore more indications. And now we are about to raise round about 30 million. And by the end of 2020, we expect to raise round about 200 million to enter into clinical trials. I thank you for your attention, and I'm ready for questions if someone have. Thank you.
Thank you, Einar. Very interesting presentation. I'm sure there are questions in the audience. Let's start with you.
BSSL is new to me. We have that enzyme for a reason. But is the situation that if you are healthy, this is not secreted? What I'm aiming at, if you inhibit this substance, doesn't that have any effect on the normal activities in the body?
That's a good question. Everybody have a background level in the bloodstream. So when this is raised, that is what we correlate to the state of inflammation. But for the instance, with the knockout mice, where you actually don't have the BSSL, you see no negative effect on that on the mice. But we are actually, you're asking a very interesting question, because we have an idea on getting an accelerated proof of concept in man that would give us that much faster than waiting for the clinical trials. And that relates actually to this question. What happens if you remove BSSL completely from the bloodstream? And what is the turnover? How fast will it be new? And that is also related to the dosing, of course. How high a dose do you need to reduce the level? But at this point, we don't believe that you need to eliminate it from the bloodstream. You just have to reduce it. And that's what we probably will aim for. But that is, of course, what clinical trials should do to tell us.
That's a very interesting question. I was actually thinking that the first question many investors would be asking is, are you going public anytime soon?
But now you ask it. Yeah, we haven't scheduled that. Absolutely not. We have, of course, thought about it, and that is a possibility, but that is not in the plan at this point of time. No.
Okay. Yeah, and so given the fact that you're not a listed company, I would presume that you could perhaps be at liberty to speak more freely than some of your colleagues we met here today. And so. The general focus, you have a partnering strategy within osteoarthritis, you mentioned, and are there any ongoing discussions?
Oh yeah. I mean, of course, when you meet big pharma, they are very interested, but most of them say, okay, it's a little bit too early. Come back when you've done that, and then you've done that, and you come back and say, oh, very interesting. But come back when you have done that. Yeah. But in the long run, you need to have a dialogue with them. To get acquainted with each other, and I think we met most of the majors. Some we definitely have in the discussion right now, and that's true also for venture capitalists. Yeah. They also need to get the message a couple of times before they understand it properly, so to speak. But as you said here in the previous question, BSSL is completely new for you. But I'm not surprised, because when we discuss this with major big pharma, they never heard about protein as a target in inflammatory diseases. And that is, of course, quite remarkable, considering that for rheumatoid arthritis alone, the market is around about $25 billion. So that is, of course, extremely unique to have such a novel target molecule.
You're bringing a new thing to the table. You mentioned a financing round. You mentioned around 20 or 30 million you were going to raise?
Yeah, roundabout, yeah.
And that's going on?
That's going on right now. That's
right
the discussion we have right now, yes.
And if any investors listening or watching this would be interested,
everybody's welcome to contact us, yes, please.
Okay, and then just go to your homepage and find your email.
I mean, the more bio you have, the better it is.
Yeah, okay. Great, thank you, Einar. We have to go further, but you will also do an interview with my colleague Linus after this. So thank you very much.